Thank you, Romain, and thank you, Matthias and Romain, for the invitation in this really fantastic place. I should have come 2 years ago and that I didn't come, so I'm very happy that you gave me a second chance to come again. So, yeah, my lab is at the Vision Institute, and the name of my group is Wavefront Engineering Microscopy Group. So I will start by presenting them. So it's an interdisciplinary lab, so we are physicists, engineers, but we also have in the team a neurophysiologist and biophysicist. And this is because we have a dual interest. From one side is to develop new optical methods for all optical circuits manipulation, and then to the more and more to use what we develop for investigating neuronal circuits, specifically since when we moved to the Vision Institute to work circuits implicated in vision, we mostly work in a mouse model. So because I'm afraid that most of what we do is a little bit far from your field of competence, so I will start with a slow introduction, so at least to make clear which are the main questions, which are also the meaning of all optical circuits manipulation. So let we really start from the beginning. What is a neuronal circuit? So in a very general way to define a neuronal circuit, we can define this as a population of neurons connected by synapses to carry out a specific function when they are activated. Okay, so this is the very general definition of a neuronal circuit. And now something that you probably know is that the most important way the neurons within a circuit have to communicate among them is through the exchange of a specific class of electrical signal that can be exchanged. Sorry, is it possible to close the door to have a little bit more? So they can be exchanged either in the form of a single spike that we call an action potentials, or in the form of a trains of spike, and of which the frequency, as you will see in the next slides, is also very important parameters. Okay, so what do we need to investigate a neuronal circuit? First of all, we need to know where the neuronal circuit is. So we have to locate the neuronal circuits in the brain. So there is something that it could be obvious for you today, is that obviously different parts of the brains will control a different behavior. But this is actually a concept that is not so old. And the very first proves that indeed if you touch different brain area, there is a different part of your body that responds. It's something actually that started around 1950, so it's not even 100 years ago. And this investigation pioneered by Wilfried Pandy consisted really with in human with electrostimulation to touch different parts, in this case of the sensory cortex, and look at which part of the brain, which part in this case of your behavior this stimulation was controlling. Okay, so the very first step is to be able to locate a circuit in the brain to understand what the circuit does. And then the second point is to understand how this circuit is able to control a function. And then we have to to decide what do we have to study in a circuit to assess how the circuits control the behavior. And again, there are concepts that for us seems to be obvious, but these are also not so old. And we start to speak about neuron and the importance of a neuron in the circuits with the study of Ramon Cajal that started to predict that really the brain is made of neurons, and also to demonstrate that there are this entity in the circuits. And so at that time, the claim was that really are the neuron the functional unit of the nervous system. And so this requires to say, okay, if I want to investigate the circuits, I need to have approaches that enable to look, to manipulate with single cell resolution, because we really want to see what happens at the level of a single cell. But then things becomes more complicated, or there have been about 50 years after the neuronal doctrine, Donald Hebb with other people also, they started to introduce the idea of cell assembly. So actually they start to say it's probably not enough to understand the complexity of the brain if we just look at one neuron at a time, but we need more to look at the brain in a more collective way. And this is because we need to individuate those cell assemblies. So the idea was that actually it's more a group of cells that are the building block of the brain function, and there are different ways to define those cell assembly, depending also in which brain area you find them. So in the cortical area, you can define the cell assembly or a collection of principal neurons that fire together, so they have a spiking activity which is highly synchronized, fire together over a time scale of tens of milliseconds. You can also define then as a coactive group of cortical neurons that you can find in spontaneous or evoked activity that mediate perception of behavior. And then if you are more in the neuroscience part that is interested in understanding memory and learning, then these cell assembly, which are also called engram, these are population of cells that build up after being modified by a series of biochemical and biophysical reactions that happens, for example, during learning or memory. And once this cell assembly is formed, they can stay a long time afterwards. And this is why you can recall the activity of this assembly, so you can have the perception of a memory, because they can be activated independently of the original stimuli that has created, that has created this assembly. And this is also something you can think of about your dream. So there are also population of cells that formed during the day experience, and then they can start to spontaneously be activated in the same way during night, and this would give you the perception of sleep, okay? The perception of a dream. Okay, cell assembly. This is a very important concept in modern neuroscience. And it seems that actually, and so what does it mean practically is that it's not only enough to look at one cell at a time, but if we want to catch the importance of the cell assembly, we need to be able to look at a larger neuronal population. So what do we need to study those cell assembly? First of all, we have to find them. So to find them means that we need to be able to record the activity of multiple cells, so that among those cells you can find if there are some of them that have a synchronous activity. So we have to be able to look to multiple cells, and then to establish where those cell assemblies are located in a specific circuit, and how many of them constitute a specific cell assembly. And then we have to be able to measure their firing patterns, so to test their degree of synchronicity. But we also want to manipulate those cells, because we want to see, it is true that once the cell assembly is formed, for example, it is true that if I just touch one of them, the entire assemblies get activated. And then what it seems that forms a cell assembly is the fact that at a certain point during development or doing a certain stimuli, those cells have a very increased, actually the connectivity among those cells becomes stronger and stronger. And so this we need to have approaches that enables to manipulate those cells, to assess how they are connected, to play back the activity that we might record, okay? And all of that, ideally, is something we want to do at cellular resolution. So how can we record and manipulate those cell assembly? Typically, this is done was done with electrophysiology. It is actually the approach for which we know that enable to know mostly all what we know of the brain today. So we can use an electrode to patch a neuron, and this enables to very precisely evoke or record the electrical activity of a single cell. So it's very precise in time and space. But the problem, for what I was saying before, if you just look at the cell at a time, you lose the complexity of the function of a circuit. So you might want to control many cells at the same time. And then you can place what we call extracellular electrodes. So these are electrodes that they can actually activate or record for multiple cells at the same time. So you get this feeling of a population activity, but the limit of this kind of approach is that it lacks single cell resolution. I mean, you can retrieve the response of a single cell resolution, but these require quite complex signaling processes, but most importantly, they lack the genetic specificity. You will understand this better afterwards. So it means that if I place an electrode, eventually all the cells that fall in the stimulated area are going to be activated, or they are going to be registered. And anyway, both cases, those approaches are invasive, because you really need to be in physical contact with the part of the brain you want to manipulate. So for years, people have looked to an alternative to electrical stimulation, and obviously the very promising approach is to replace electrode with light, so that we can ideally be able to read and write, so to register, to image, and manipulate neuronal cells without the use of electrode. And this is something today we can do. We can actually image, record neuronal activity using mostly genetically encoded calcium and voltage indicator, but we can also manipulate neuronal activity using optogenetics. And so this is what I will now try to develop more. So first, on the reading side, so if we want to image, to record an electrical signal with an optical microscope, the first things to be done is to be able to convert this electrical signal, this spike, into an optical signal. And this can be done by, for example, using a genetically encoded calcium, and we will see afterwards indicator, a voltage indicator. So how do they work? So these are fluorescent proteins that has been engineered so to become brighter when they bound to a calcium ion. Something you should know, or probably you know already, is that each time you have an electrical signal propagating through a cell, this changes the membrane potential, and this opens some, among others, some calcium selective channel, so that each time you have an electrical signal propagating through a neuron, you have an massive entrance of calcium in the interior of the cells. And so now if you can, and we will see how later on, if you can put those proteins in the cell, at the interiors of the cell, each time you have electrical signal propagating, you will see that the fluorescent signal becomes brighter. I will show you this in this movie, which is one of the first one that was done to show how those indicator works. What you will see here is in red, you will see the electrical signal measured with a patch pipette, and in yellow you will see the corresponding fluorescent signal. So here we are. So you see there is a cell. Here you should see there is an electrode. So in red, I don't know if you can see it. In red you see the spike, and in yellow you see the corresponding fluorescent signal. Most of those indicators are highly nonlinear, so you see when you have a burst, you have a very huge fluorescent signal. Were you able to see it? Okay. So, and there is another class of probes that we can use to probe, which are called genetically encoded voltage indicator. We will come more on the comparison among those two probes later on, but let me just say quickly, there are multiple of them which are based on slightly different principles. Here I just selected the scheme for the GFP-based voltage indicator. So again, these are fluorescent protein that has been modified to be bound to a voltage-sensitive domain. They are again expressed, in this case, they are expressed on the cell membrane. And what happens is that, again, when the action potential propagates, this changes the membrane potential, and this change can either quench or enhance the fluorescence of your fluorescent domain. And so now we are not sensitive to the calcium, but we are really sensitive to what change, to the membrane potential change. And here you will see another example where this was, you see that there is this blinking, and so this blinking is each time you see a change in fluorescence, there is a change in the membrane potential. What you can already see here, but we will come better on this point later on, those probes are much faster, the calcium indicator, so actually you can have the temporal resolution to resolve a single spike, which you could not do with calcium imaging. But we will come to this point later on. So now, before transitioning into the rest, I wanted to specify, because we will use this also for optogenetics, what do I mean with genetically encoded voltage indicator? So how do we put those fluorophore in the cell, and why we call this expression genetically encoded? So we use virus. So the first step is to find which is the gene coding for the specific proteins. A calcium indicator, a voltage indicator, you will see afterwards the optogenetic protein. And this gene is bounded to a promoter. And then, okay, now you have this genetic sequence. Then the next step is to take some virus and to replace the viral genome with this genetic sequence by leaving the terminal that repeats for the replication and encapsulation of the virus. Now you have this engineer modified virus. You can inject them in the mice or whatever model you are working on. So the virus, because it's made for, is going to infect different neurons. And at this point, only the neurons that are able to recognize the promoter will be actually able to express the protein that is encoded by the gene. So why this is very important? Because as you can imagine, in the neurons, in the brain, even if you choose just to look in a small area, there are thousands of neurons, and there are many cell type. And so if you want to just study a specific cell type with this genetic targeting, you can make only the cell of interest become light sensitive, okay? So we will come to this afterwards also for optogenetics. Okay, so now the goal, say, we have those proteins respond to light, and they enable us to visualize the propagation of an electrical signal. But we want to look at those electrical signals in scattering medium, so we want to be able to resolve the activity eventually at the resolution of a single cell when we look in a scattering medium, as is the brain. And so we need to use a specific kind of light excitation, which is two-photon excitation. This, I guess, most of you know, so I will go relatively fast. But I mean, the idea of two-photon microscopy, a process that has been predicted many years ago by Maria Goppemeyer, is that a molecule or an atom can be equally excited by the absorption of a single photon, but also by the quasi-simultaneous absorption of two photons of roughly the half of the energy, going through these virtual states. So why this effect is very important in microscopy? There are two main advantages. The first of all, that you can excite the same kind of molecules by using longer wavelength light. So as you well know, especially in this school, this allows you to penetrate more because of the scattering. And the other important characteristic of two-photon microscopy is that because for the excitation you need this double jump, so the absorption probability will depend on the square of the excitation density. So the more the photons are spatially concentrated, the higher is the probability of doing this double jump. And so now if you well focalize the light down to a small spot, eventually only the targets that lie exactly at the focal point will be excited, while you have a very nice rejected of out-of-focus light, which is not the case of single photon microscopy. Also, because this effect is very unlikely, it's also important to concentrate the photon temporally. So in two-photon microscopy, what you typically use is femtosecond laser, so that you not only concentrate the photon spatially, but also temporally, to increase even more the probability of this two-photon absorption. We will come to this point afterwards. Okay, so now what you can do, you take your microscope, you have this small spot, the spot is scanned on a certain brain of injured, on a certain circuit, and then you can retrieve the fluorescence from your sample, and this is a optically confined image, so basically only the cell which are right at the focal plane will contribute to your signal. But now interestingly, you can keep on repeating this scanning. And so in your preparation, if you have put those calcium indicator, you not only will see a static image, but you will see something dynamics. So this is an example of an image that has been done on the visual cortex of mouse, while there were different grids that are projected to the animal, and you see that we see this blinking, and now you know that each time we see something becoming brighter is because there is a spike of a burst of spike that is produced by this cell. So calcium imaging is really one of the most adopted approach today to look in intact brain, and there are many things that we learned thanks to calcium imaging. Here I will show you just few things because these are related to the rest of the talk. So the one properties that was possible indeed to prove with calcium imaging is the neuronal tuning. So this is the properties that the cells have to respond selectively to a specific stimuli. For example, in the visual cortex, it is known that, I mean, all the cells that you see here are all the same type, but nevertheless, at a certain point during development, something happened so that those cells start to differentiate. And for example, if we project grids of different orientation, not of them will respond in the same way, but there are some cells here in yellow that will be fired when the grids has this orientation, other when the orientation is this one, other one when this is one. And this is what we call the neuronal tuning. Okay, so within a population of cell, at a certain point, something happens, so their functional response become tuned to a specific stimuli. This can be the visual cortex, but you can find those kind of properties in many other brain regions, so you can find cells that selectively respond to a different sounds or to a different odor, okay? So, and this is also an example of those cell assembly I was pinging you before. So there are cells that at a certain point become highly coordinated to select for a to give a response to a specific stimuli. There is something also the brain region where neuronal tuning is very fascinating is in the hippocampus. So here also there are cells that are tuned, and specifically there are different kinds of cells that encode for the location, speed, and direction. These are the grid cell, the place cell, the border cell. And so I will show you this movie here. This is with electrophysiology, but this is spectacular. And you will see here now the mice is moving in a cage. And there are some electrode placed into the hippocampus, so a very deep brain region, which has nothing to do with vision. And despite of that, the orientation in the space is mostly encoded by this brain region. And you will see now that by looking at how the activity of those 10 different cells, which are encoded with 10 different colors, you will see that each of these cells fire in a specific position of the cage. When the mice the rat actually is in a specific position of the cage. You see, so this cell is mostly firing when it's in the center, the blue one when it's there, and so on. And so if you repeat this experience, you will find that at the end, there is really, by those 10 cells that have a specific location where they will fire. So there is something in the brain that tells to the rat where he is in the space. And this kind of experience has been then repeated by also using calcium imaging in mice, so you can refine the temporal and spatial resolution, and actually the Moser couple, it got the Nobel Prize for finding and demonstrate the presence of those cells in the hippocampus. Okay, so we can see, we can image, we can prove that there are cell assemblies, we can prove that there are cells which have a different functional role. But now, if you want to understand how they work, as I was saying before, it's not enough just to watch what happens. We might want to manipulate those cells because we want to understand how they are connected. We want to understand what happens if I change the activity of just one of these assembly. How many of these cells need to really be controlled to recreate the perception of a certain perception, or to even modify a certain behavior. So we want to do this again using light. And this is the revolution of optogenetics, which probably some of you have already heard about this. So again, the starting point is to find some specific proteins in nature. These are called the microbial opsins, which are photosensitive channel or pump that can be also expressed in this genetically way on the cell membrane. And so now when you send the light, you can, the absorption of light induce a conformational change in the protein. So it's as if you can open a very small hole, and this gives the entrance of positive or negative ions, which is the same that you will do with an electrode, okay? So you will produce a current of a specific sign, and this current will be able to evoke an action potential. So you see in correspondence of each light pulse, you will have a spike. But you can also inhibit the propagation of the signal if you use the inhibitory opsin. So they are also expressed in a genetic way, so you can specifically define which cell type is going to express the protein, sorry. And so in this is one of the very first movie where people have shown the optogenetics. In this animal, they have expressed an activatory opsin in the mouse cortex, in the motor cortex, sorry, and there is an optical fiber that you cannot see but is there, and which brings light into the mouse head and on the right part of the motor cortex, which control the movement toward the left. And so now what you will see here is that indeed, so then the mice at the beginning has a normal behavior. When the light is on, that you may see there is some blue light here, he starts to move toward the left. Okay, so we do have a specific control of a specific behavior by sending light in a genetically identified cell population. Okay, so this, what I'm showing here, is basically one of the most incredible application of optogenetics, and this is the fact to giving you the tool to really start to do a mapping of brain function. So we have seen that this kind of investigation was started by Panfield many years ago, but at that time, what people needed was to really do a massive electric stimulation of specific brain area and see the effect. Now we can do much more. First of all, we don't need anymore to have this invasive approach. This can be done just with light. And plus, we can not only correlate a specific behavior with a specific brain region, but we can start to correlate a behavior with a specific cell type. Okay, so this is the huge revolution of optogenetics, mapping brain function with cell type specificity. And so there is a very large number of works. Well, with this approach, people have started, for example, to start to find the neurons involved in memory, in fear, anxiety, addiction, depression, and so on. And in this kind of study, the approach is very similar to the one of the movies I showed you before. So you have an optical fiber, you shine blue light in a relatively large brain region. You have identified with your molecular probes which is the cell type that is going to react to light. And now if you see something, you say, okay, this cell type is doing what I'm seeing. Fantastic approach, very important, but there is a limitation in this kind of optical approach, is that, as you can see here, if I send light in a large population of cells which are all photosensitive, I will eventually make all these cells firing in a synchronous way, but all the cells that are reached with light. And now if you remember, and I will show you another movie here, if you remember what we would see if we could watch in a circuit in a physiological condition, it's very rare that all the cells in your field of view will fire in a synchronous way. You will have at most a small group of cells, but in general, I mean, each element, okay, yeah, you can see it. Each cell has a very independent activity, okay? So each cell or cell assembly contributed in activity of a large circuit with a very specific special temporal signature. So if we want to start to play with this, if we want to start to play back this kind of activity, if we want to modify what we see to see the effect on the behavior, or to see the effect on another cell population, we need to replace this whole region optogenetics with something more precise that we term the circuits optogenetics, meaning with this a combination of approaches that enables you to manipulate a single cell or a specific group of cells independently in space and time. Okay, so and we want to do this with cellular resolution. We want to do this in scattering medium. So what we need for that, we need to develop approaches that enable us to control or to inhibit the activity of a single cell or a specific group of cells with millisecond temporal resolution. Eventually, in multiple planes, you will know the brain is not just a flat area, and if possible, in corresponding with freely moving mice, so to be also have access to the manipulation of circuits that control behaviors that require freely moving. I don't know if we can go through all up to freely moving. Let's see how long it will take, but let's start with the first point. So we want to use light to manipulate single of multiple cells at single cell resolution in scattering medium. So again, the first step is to replace this wide field unspecific light illumination with two-photon excitation. But what it turned out that with optogenetics is not so straightforward that as it was for imaging, because if you use a conventional two-photon microscope, in most of the case, this very small illumination volume that is used in imaging is not enough to open enough channel to evoke an action potential or to keep a cell under inhibition. And the second point is that if you want to manipulate a cell assembly, if you want to manipulate multiple cells at the same time, and we go for conventional scanning microscopy, we won't have the time to control a cell assembly within a millisecond temporal scale. And so at the end, what it turned out that using two-photon for optogenetics requires to find approach that from one side enable to illuminate more channel at the same time, and also to multiplex the light at multiple targets, so we can eventually at the same time control the activity of multiple cells. Okay, some physics now. So how can we do this? So we have worked in two approaches, computer-generated holography and generalized phase contrast method, so to be able to precisely shape the light, so that instead of just covering a single just illuminate with a single spot, we can eventually illuminate the entire cells. So you have already heard about spatial light modulator. We can modulate the wavefront. And this can be done with these two approaches. So I will now quickly go through them. So computer-generated holography is the modern version of holography that you probably all knows, but just to remind you how it works. So the idea of holography was to be able to reproduce with light a certain object in absence of the object. And the system is not very far from what was already schematized by Dennis Gabor, which is the one that proposed holography. So you need a laser, and you need to divide the laser in two beams, a reference beam that is just propagating, and then another beam which is illuminating the object that you would like eventually to recreate with light. This beam going through the object is going to be diffracted, and then you let these diffracted waves to propagate, and at a certain plane you can record the interferogram between the reference and the diffracted beams. And now when you have this interferogram, what you can just use your reference beam to illuminate your interferogram, and this will create in a real and virtual space the shape of the object. Okay, so this is holography. It has been largely used in physics. It has been largely used in science fiction movie where people start to dream, okay, with light we can eventually create a three-dimensional object and also to animate this object in time. So today, with computer-generated holography, we are not so far from this science fiction prediction where we can indeed replace the original holographic system where now you don't need anymore physically to have an object in your light path. You can just draw it by computer, so you can start with an image template. And we don't need to physically record the interferogram. This can be calculated with Fourier transform-based algorithm. So you calculate your phase hologram, and then this can be sent on a liquid crystal screen, which is a dynamic device. So you can continuously refresh your phase hologram and generate a different phase object. And if things are well done, your laser, which is now your reference beam, going through the spatial light modulator, is creating your shapes. Okay. So the point is that we wanted to use this light to precisely illuminate a neuron in a neuronal circuit. So the point was, can we reach a so precise light propagation control so that we can really send the light only when we want to send the light. And so the first point was to know where we want to send the light. So for that, what we propose is to simply start with the fluorescence. Okay, so you take your fluorescence image of your sample, and then on the base of the fluorescence, you decide, okay, I want to illuminate only this dendritic part, or among this population, I want only to illuminate those cells here. So you take this part of an image, you calculate the phase hologram, and when the light is going through, is eventually creating this illumination spot, which is not anymore this small spot that we use into photon imaging, but is now a shaped distribution of light. And obviously, you can also simply create disk, or you can simply create a multiple point. We will come to this point also later on. As I was saying before, the huge advantage of holography is that you can, the huge advantage of using spatial light modulator is that you can send multiple phase hologram one after the other. So not only create a static light distribution, but you can generate an animated function, and which also in a sort of 3D shaping. Okay, so we have precision, we have the possibility of having a dynamic light distribution. This is one approach. The other one is generalized phase contrast. I will quickly talk to you about also this one because it's something we will use at the very end of the lecture if I can manage to reach that point. So generalized phase contrast is also a modern version of a very old approach in physics, phase contrast microscopy. This time, this inventor is Frederick Zernick. So phase contrast microscopy is something that was introduced in order to be able to visualize a transparent object. And so, for example, here, I don't know if you can see that this is just a cover slip with few cells, and it's very difficult to look at those cells because they are very thin object. And so if you simply, so you can represent the transmission of this screen with a term which represented the phase change that you will impose to the light going through, and with a certain special distribution. Now, if you send the light, if these are a very thin object, you can approximate this transmission function with a Taylor expansion, so you will have a constant term and a special position-dependent phase modulation. And now, when the light is going through this screen, you will have, on the other side, you will have an undiffracted component, so most of the light actually will continue to propagate as it was, and there is a very small part of the light that will keep up the phase shift imposed by the cells. But now when you look at the intensity distribution, because this phase shift is very small, most of the light is just undiffracted light, and this is why it's very difficult to see the contrast in this kind of imaging. And so the idea was, can we increase the contrast? And the idea of Ozernik was to take advantage of the fact that the undiffracted light is has a non-axis propagation, while the diffracted lights go somewhere else. So physically, if you put a lens, they're going to be specially focalized in different planes. So the undiffracted light is focalized exactly at the focal point of the lens. The rest will be focalized around. So you can specially separate the on-axis from the off-axis component. And so the idea was, okay, what about now if I do something on the on-axis component so that I can make those two components to interfere? And so the idea was simply to put a phase disk, so that you can introduce into the on-axis component a pi/2 phase shift. And if you do this, when you are doing the same and you look at the intensity distribution, you will see that the light transmitted through this cover slip after this phase modulation will be given by a constant part, so again this undiffracted component, plus a modulated term that now spatially is distributed as it was the distribution of the phase object. And so you can really convert this image into that one, so you have increased the contrast. Okay, this is phase contrast microscopy. What do we do with this with optogenetics? So a few years after, it was Jesper Gluckstadt that did diffuse the formalism for what he called the generalized phase contrast method, finding the condition that for which you can build up this interferogram for phase object of arbitrary, imposing an arbitrary phase shift. So we are not anymore related to having this small phase shift, but this can be as big as you want. And the second point is now our phase object is not in the sample plane, but is actually a special A modulator. So we can impose any phase distribution into the special A modulator, but with the same physics I was describing before, this phase object is transformed into an intensity modulation. Okay, so we can actually transform into an intensity modulation any phase distribution I send on the special A modulator. And then you can find the conditions. So now the phase shift has to be of pi, and if you well do the things, you can have a maximum contrast. So we have intensity in the region that you want to pattern, and you have black everywhere else. So again, you can use this for light patterning. So now somehow is easier than holography. So you take your image, and now you decide the region that you want to illuminate. This is now your phase pattern. So this is not anymore this complex hologram. It's really a binary the conversion of this intensity into a phase shift, and this is what you get at the sample plane. So very precise light distribution, and you can also play with the phase level, and so to generate also a graded intensity distribution. Okay, so both approach in different situations that I will make clear later on can be used to have this very precise light distribution on the sample, so that you can very precisely illuminate the region you want to. But now, as you know, in optics, each time we make a larger light distribution on the plane, you also pay the price in the axial resolution, right? So with generalized phase contrast method, this is a drastic change. So the larger is the spot, the worse is the axial resolution. The dependence is to the square. So the axial resolution is proportional to the square of the spot. So if you want to cover a neuron of about 10 micrometers, you will have a column of about 100 micrometers. With holography, it can be shown that the situation is slightly better, but so you have a linear dependence from the axial to the lateral spot size, which is fine if you want to just target a single neuron, because actually you will have a resolution of few micrometers. But if you now you want to illuminate a group of cells which are close each other, you will see that actually there is a sort of a very long light distribution, and so you completely lose the capability for single cell resolution. So if you wanted to use this shaped approach to get single cell resolution, we need to do something more so that we could go back to the axial resolution typical of two-photon microscopy. And so for that, we propose to combine those two approaches with the approach of temporal focusing. I know if any of you know about this approach. So this has been pioneered by the group of Danoron, and the idea of temporal focusing is the following. If you remember, two-photon signal is depend on the excitation density, so this is why we use a strongly focalized point, but also depends on the pulse duration. So we need to use pulsed laser because the more the photons are temporally concentrated, the higher is the probability of the two-photon absorption, okay? So the shorter is the pulse, the more signal you get, and the larger is the pulse, the less signal you get. So the idea of temporal focusing was the following. We use a dispersive grading. As you well know, in a pulsed laser, you have a certain bandwidth, so you have a few colors. And now if you send your beam through gratings, the different color will be diffracted through slightly different angles. And so this will introduce a difference in the optical path of the different colors, which is more important the more you move away from the grating. And so this, as you probably will know, that the more you introduce a difference in the optical path, the broader will become the pulse, okay? So now if the grading is conjugated with the sample plane, nothing will happen to the original pulse. So at the focal plane, you have a very nice pulse, a very short, nice pulse. But when you move out of the focal plane, your pulse will become broader and broader. And because of this dependence here, the absorption will be enhanced at the focal plane, and you can nicely reject the out-of-focus light independent of having or not a focalized spot, okay? If this was not clear, you can also see everything in the temporal domain, which for me is easier. But so forget about colors and all what I was saying, and now just try to see your beam as made of pulse. Okay, so during the time that your beam, you will have some disk that will propagate, and now what you will see here is what happens at this beam. So when your pulse is propagating, now you have to imagine it's a big pulse because we want a large illumination shape. So this disk is propagating, it meets the grating. So at the beginning, the intersection between this pulse and the grating will be at the first part of your disk. And then the more the pulse propagates, the more the intersection will move in this direction, okay? Which practically means that it's as if your grating is going to be illuminated with a very fast scanning line. You can probably visualize this better here. Okay, so this is the pulse, this is the grating. And what you will see, okay, now when we move the grating, this is what will happen if you could watch on the grating, okay? And this is the same what would happen on the sample because the sample is conjugated with the grating. I don't know if this was clear. So anyway, so you have this disk, the intersection of a disk with a plane is a line, and because the propagation is at the speed of light, this line will very quickly scan the sample. So why this is very important? Because this means that even if you have a large spot which is illuminating a large area, physically, this area is never going to be illuminated at the same time, but there will be only a line. And so the axial resolution is not the one of produced by a large spot, but is the one produced by a line, okay? So we wanted to do temporal focusing and light shaping, so this is just a technical detail. So before reaching the grating, we have a special light modulator, so that the grating is illuminated with a disk or with this distribution of disk. And now, because we have temporal focusing, the axial resolution is nicely concentrated around the focal plane. And what is interesting is now this with generalized phase contrast works even better. So we have a very, very sharp absorption only at the focal plane. And interesting now, what happens is that no matter how large is the light distribution on the sample, you will always get the same axial resolution. So you have somehow decoupled the lateral from the axial resolution. Okay, so years ago, we demonstrated for the first time that the combination of this approach is really enabled to do two-photon optogenetics. And then this was in brain slides, and then you can do this in vivo. You can also show that you can reproduce the same spiking pattern with a sub-millisecond temporal precision. We will see why this is important afterwards. And it's also possible to control the spiking rate, so to generate a spike with up to 100 Hertz. These data are in vivo. And now just to show you something to give you a perception of how precise we can be with all these approaches. Now, what you will see in this movie, these are cells, visual cortex in vivo mouse, layer 2-3. In red, you will see the cells. And now in those cells, we have a fluorescent reporter, which is the one that enables us to find the cell, and so to design, to draw our hologram. But in these cells, you also have an opsin, so they become light sensitive. And there is also a calcium indicator. So we can activate the opsin, but also look at the effect of our activation. So if we are really producing spike, we should see an increase of the calcium response. And this is what you will see here now. Yeah. I don't know if you see actually, but okay, yes, you probably see. So here we put 3 spots, so an hologram producing 3 spots. The 3 cells are nicely responding, okay? And now we want to activate only one of them, so I only have an hologram generating a single pole, only that cell is responding. So you have to consider those cells as 10-20 micrometer, the separation is 10-20 micrometers, so the approach, and this is in vivo, so the approach is really robust and precise. Okay. Very quickly to say, just to say, we can activate, so we can produce spike, we can also inhibit, we have just to change the opsin, so it's also possible to use the photon excitation, so to stop the propagation of a signal. And there are also now new viruses that express in the same cells an inhibitory and an excitatory opsin, so by changing the wavelength, we can either activate or inhibit the neurons, and this is very important to understand the role of specific neuron in a circuits, what happens when I activate or inhibit that neuron. Okay, so before going on with a few other technical modifications we did on the holographic system, I wanted to show you a couple of examples to really show that indeed the precision that we take a long time to really reach the precision for single cell resolution, and to show you that this is really important, at least for few applications. So the first one is a collaboration we have with a group of Rosa Cossatti in Marseille. And the interest of our lab is really to show that there are those hub neurons. So it's really to show that in a circuit, not all the cells have the same role, but there are cells that are more important than the other. And so there are cells that alone can orchestrate the activity of the entire circuits. They were able to demonstrate those cells in vitro with electrophysiology, but the point was, can we watch what those neurons do when we look in vivo? And so the experiment is the following. Again, in the cell, we have a calcium indicator, so we can look at what watch what the circuit is doing. We have an opsin, so we can alterate or manipulate the activity of those cells. So the first part of the experiment, which is also the first part of the movie, you see, is simply to watch what happens in the circuits. These are developing circuits, so this is why you see this very high frequency activity, because really cells are continuously active and they are forming the synapses, they are formed what one day will become the cell assembly. And so from this image, they could retrieve what we call the functional connectivity analysis. So by looking at the activity of each cell and how this activity is correlated or not with the rest of the cells of the circuits, they can find that there are some cells which are highly correlated with all the rest. Okay, each time those cells spike, there is a very high number of cells that follow. And so these are those putative hub cells. So probably these are really the cells that have a much more important role than, for example, this cell here. Okay, so the size of the disk is proportional to the connectivity probability. And so now the question was, what happens if I just modify the activity of one of those cells, which in this film is this one. So you will see now that at a certain point we place an holographic spot here. And then they kept on doing calcium imaging. Okay, so again, you see now, I don't know where. So now this is just the activity. Now we put the spot. And you see that when we alter the activity of this cell, there is a global decrease of the average activity that you can see from here, that there is an increase in the inter-event interval. And then at a certain point, we will remove the spot from the cell. And what you will see Okay, the network start to fire again as it was before. Okay, so this was a very important result because it was the first time that in vivo was possible really to demonstrate that just a single cell can have a major impact on an entire microcircuits. There is another class of experiment where this precision of single cell resolution is very important, is for connectivity. So I hope I more or less convince you that when you want to understand how circuits work, what is very important, one very major property of the circuits, how cells are connected. Okay, so this enables you to understand, for example, what make a cell assembly is because they are highly connected. With this tuning probability, what it's come from is because they receive certain input, or is simply because they are highly much more connected among them with respect to the other cell. So how can we measure connectivity? So how can we measure if two objects are connected? So the most obvious thing is to say, okay, I look at the two of them, I start to manipulate one of them, and I see if the other one is following. If this is the case, the two cells are connected. So normally this was done with electrophysiology. So you can put an electrode, generate a spike on what we call the presynaptic cells. So this at this point is going to spike. And at the same time, we record the activity of the postsynaptic cells. If we see something, it means that the two cells are connected. If not, they are not connected. Now, you can see from this cartoon that you require not only to patch one cell, but two of them. And then if you want to probe multiple cells, you need to move the electrode on the presynaptic cells around, and at a certain point, unfortunately, your postsynaptic cell will die. So the length of this experiment depends on the time life of your postsynaptic cells. And so it was possible with this approach to measure 4 to 12 connections per experiment. Okay, now the brain is made of many, many cells, so this is a very small number with respect to what you need to really understand how the circuits work. So there was a big improvement in this approach when optogenetics came into play, so even more when single-cell optogenetics comes into play. So now, instead of placing an electron on the presynaptic cells, we can just send light and then move your illumination spot around. And so from 4 to 12 connections is now possible to probe hundreds of connections. Okay, so this is already a big jump in the field. Sorry, how much time do I have? Okay, so we wanted to see whether this was possible to be done in vivo, because ideally you want to functionally find the cell assembly. For example, for this you need to work in vivo, and then you want to see how they are connected. So in vivo is even more complicated because there was no way to do this experiment in electrophysiology. And so the experiment is the following. So these are again visual cortex. You visualize the cells in the field of view as I was doing before. Then we still need an electrode on the postsynaptic cells. And now what you can do is on the base of these numbers, of the image, you can retrieve the coordinates of all your possible presynaptic cells and now generate a series of holograms. Each holograms will send a spot on a specific location. And at each illumination, you will record what happens on the postsynaptic cell. Nothing here, so those two are not connected, nothing there, not connected. A response, the two are connected, okay? So already simply by changing electrode with light in vivo, it was possible. I want to tell you this is a very, very small signal, so this experiment has to be repeated 30 times to really extract the real signal. But still, it was possible to probe hundreds of cells. We wanted to try to speed up the process even more, so to have access to even more cells, and we know that with holography we can not only illuminate one spot at a time, but we can also generate holograms that illuminate a certain number of spots at a time. So this will speed up the process, right? So now we have a series of holograms. Each of them is going to illuminate a randomly choose subset of cells. Okay, and again, we do the same experiment. Nothing here, so none of those cells is connected. Nothing there, none of those cells is connected. Here, something is happening, okay? So within this ensemble of cells, there is one of all of them that are connected because I see a response. Okay, now the question is, when you do this, how do we find back within those cells which are connected. So you have two options. Either now you just go sequential light stimulation of this subset of cells to find the one that are connected, or you can apply some compression sensing analysis, which I will try to explain you here. So these are the real data. This is an experiment where, imagine now we have, in this specific field of view, we have 43 cells, and in sequential holographic stimulation, you have to do 43 experiments and repeat it 30 times. And at a certain point, you start to see, okay, there something is happening. Okay, so that cell is connected. Now I want to speed up the process. So instead of doing 43 experiments, we just do 15 experiments by sending 15 holograms that activate each of them 7 cells. So obviously you reduce the number of experiments. And now there are 4 metrics that give you a response. So how can we find back that one was the cell? So here you have a system now where you have a certain number of measurements, which is in this case, if you do the 43, okay, a certain number of neurons, 43, and now you can either use the same number of cell experiment and the same number of unknown parameter. Fine, this is what we do here. But if you can also reduce the number of cells, so you can of experiments, so you have a number of metrics where you see in black is the cell that are activated. This is an undetermined system, so in principle you can find a solution. But in practice, this is exactly the same that you would do in compression sensing for imaging. There are situations where when you have a sparsity, which is the case in this case, so you have a low probability of connection, you can and you have also in the condition of a linear system, meaning that if we have 2 cells that are connected, when they are 2 of them activated, they will produce the sum of the response. So in this condition, sparse and linear system, you can apply the compression sensing analysis, and I have to speed up a little bit on that, but indeed it is possible to retrieve the connected cells. So we did a number of metrics as big as the cells, and then we start to see to which extent we can stop and find a good response, and we found that up to we can retrieve the connected cells with a compressed ratio of 3. So what that means that you can either reduce of 3 the number of experiments that you do, or you can increase up by 3 the number of cells that you probe. Okay, so we are really on the way to now probe hundreds of connection in vivo, which was really something that was not possible before. And you understand that for this kind of experiment, you do to have a true single cell resolution, because otherwise you won't know which cell is connected. Okay. Okay. So the very last, this part is quicker, is just to show you that one major advantage of holography is that you can not only probe and illuminate or control activity in a plane, but you can have access at targets which are in 3D, and which is what you want to increase the volume of the circuits you want to probe to really mimic physiological activity pattern. And so the question is, can we do 3D? So holography is done for that. So we can indeed generate 3-dimensional holograms. So this is just now a zeta stack where we see that we can actually shape the light in different planes. Okay, fine. This is known as holography. The problem is that we wanted also, if you remember, to apply temporal focusing. Okay. So there is a problem if you use 3-dimensional holography, is that eventually not only one of those planes will coincide with the grating for temporal focusing, and so only one of these planes will be temporally focused, and which is not what we want. So we have to find a solution, and what we found is to have a second special A modulator. So I will show you the principle here. So you will use a first special A modulator to create your disk, or to create your shape, typically just a disk of the size of the neuron you want to illuminate. And then this goes to the grating, so it's going to be temporally focused. And then you have a second special A modulator, which alone will only produce this multi-point distribution that I showed you at the very beginning. Okay, so with this SLM, you can send the light a specific point, and which are centered at the location of the cells that you want to illuminate. And now when you have the 2 of them, here you will have a convolution of this light distribution with that one. And so indeed you will have the generation of 3-dimensional temporally focused spot. Okay, I will show you an example here. Okay, so here we have 50 temporally focused disks in a large volume. All of them is the exact replica of the same, so they are all nicely axially focused. So we have now the access to hundreds of cells in a millimeter cubic volume. Okay, this is just a technical details. The system can be simplified if you don't want to use two special modulators. The first one, because it's only used to produce a disk, can be replaced with a fixed phase mask. And this approach is also consistent with other approach. So actually, we can also, instead of using holography here, using the generalized phase contrast method. And this is a way to do three-dimensional generalized phase contrast, which is otherwise not possible with the conventional GPC approach. Can also replace with Gaussian beam, can also be replaced with more complex phase amplitude modulation. But just to say that no matter what you do here, at the gradient for temporal focusing, this can multiplex it in 3D by using the second special ray modulator. So I have a few examples also on that. Where do we do 3D? But probably I will skip the first one because I want to show you the last part. Okay, so 3D is interesting not only because we want to reach multiple cells in 3D, but also because we want eventually to decouple the imaging from the photostimulation plane. So you can photostimulate in a plane and perform calcium imaging in another plane. We did this on the retina, but I won't go through that. So here the idea was to manipulate the bipolar cell layer and look what happens at the ganglion cell layer. We can see this if someone is interested later on. I wanted more to go to that because we need this for the second part. So what can we use the holography? Can we really use to play with those cell assembly? Okay, so these are, there have been a number of spectacular papers that came out in the past few years using holography to really start to probe the threshold for perception. Okay, so the schematic is, there are two very similar papers that came out, not from my lab, unfortunately, where the idea was the following. So there were mouse that has been trained to recognize the orientation of a grid. So the mouse was learned to recognize the grids in this orientation or in that one. And so according to the grids you were sending, the mouse was expected to do a certain task. In this case, to leak water right or left. So now you know that your mouse is able to recognize a grids like this or like that. At the same time, you perform calcium imaging. So you are able to see which number of cells are active while the mice is looking at these grids or to another grids. Fine. So now we found that the cell assembly responsible for the perception of the grids. And now the next step was, okay, now let's go back with holography. So we remove the grids and we activate those cells with holography. So is the mouse able to perform the correct task? And incredibly, it was the case. Okay, so it was possible to have the right response by re-illuminating back the cell that in response to a certain grids were active. And interestingly, in this paper, they show that actually once you have found your assembly, you don't even have to activate all of them. It's enough to activate, in this case, 2 of them. And because this assembly formed, so 2 of them are enough to reactivate the entire assembly, and the mouse have the right perception. So this was really spectacular results that came out in last year. And so the proof was that indeed the activation of a very few cells is enabled to trigger the activity of this cell assembly or this assembly activity. And more or less on the same line, and I need this to introduce the last part of the talk. This was another experiment in the olfactory bulb. Here the question was slightly different. The question here was, which is the minimum number, sorry, something happened here, which is the minimum number of cells that we need to control to do a specific perception, in this case olfactory perception. So again, the mouse was trained to recognize a certain odor. And then the point was, okay, now these are the cells that I see to fire when there is a certain odor coming. And so now how many of them I have to play back to have the right response from the mouse? And so they start to increase the number of cells, and you see when you are around 25, 30 cells, we have the right response, okay? Okay, this is important because it was important to establish that these in particular circuits, which is the critical number of cells you need to control to generate a certain response. But what is very important in this paper is something that was missing in the one before, is that they start to play with the time. So if you remember the very slides where I was speaking about cell assembly, so the original definition of cell assemblies were cells that fire together, wire together. So the firing time is also very important. It's not only where they are, but it's also how synchronous is their activity. And so with this, in this paper, they started to play with this. So they say, okay, now I know that I need 30 cells to have the right response. But how synchronized those 30 cells have to be activated to give the right response? So what they did, they use holography, and they use they say, okay, I can either generate an hologram that activate the 30 cells at the same time, or I can now generate 2 holograms or 3, so that instead of being activated all at the same time, they are activated with a certain temporal shift. Okay, the minimum temporal shift they could do was the 20 millisecond, the 20 millisecond refresh rate of the SLM. And actually, what they saw is that was enough to desynchronize the 20 cells of 20 milliseconds, and the response was not correct. Okay, so it's not only the number that matters, but also how synchronously they are activated. And so this brings us to the last part. And this is, can we, now that we know that we can be very precise in space, can we start to play with the temporal code, with the time code, or whatever people call it? So can we start to play also on the firing time? So the ideal experiment one day will be to be able to watch what happens in the circuits. We know that oops, sorry. We know that the cell, okay, now I crashed it. We know that the cell have this firing activity, and so can we exactly play back what we see? So can we exactly play back the sequence we see? And can we then start to slightly desynchronize what we see in a very precise way to see at which point the network is not doing anymore what was doing under physiological condition? So, which we can say, can we start to understand how close in time neurons fire together to wire together? So there are 2 technological limitations for this dreaming experiment. The first one is how fast we can perform sequential light targeting. And there are the first technical limitation is the liquid crystal refreshing rate. Okay, so if I want to send multiple holograms, there is a limited time from one to another, which is related to the time the liquid crystal takes to reorient, which is between 60 to, I would say, more 300 Hertz. And also the problem is also you have to illuminate. So you not only have to send a series of holograms, you have to also send an hologram, generate an activation pattern, move to the next. And this illumination time depends on what you want to, is from 2 to 100 milliseconds. So we want to solve this first issue first, and what we found is that in this configuration that I showed you before, where there is a first modulation approach that goes to a second special layer modulator that only produce multiple points, we actually don't need so many pixels to just generate the multiple points. So much less pixel if you want to use the SLM also to shape the light. And so why this is interesting, because actually what you can do, instead of sending a single hologram that produces your multiple point, you can send a series of holograms vertically aligned. Each hologram is encoding for a different distribution of point. And now you can use a galvanometric mirror, so that your temporal focusing light, and now can we move through the different holograms. And so this allows you to go to up to the kilohertz rate, no matter how slow is your spatial layer modulator. And so then with some other trick on the illumination, but I won't have time to go through it. But so we are close to this dreaming experiment, at least for the photostimulation time. Indeed, we can generate light stimulation at the kilohertz rate. There are some side advantage of this approach is also that you can reach square root of h time more cells, where h is the number of holograms, and importantly reduce the sample heating, something we can discuss afterwards. But okay, this is the illumination. So we are there. We do have the technology to do ultra-fast sequential light targeting. But now what we are missing is the precision in the imaging. Okay, if I want to produce this very precise pattern, I also need to see what is happening with the same temporal resolution. And then, if you remember, we have two options to watch what happens in the circuits. One is the calcium, using calcium imaging, and the other one is using voltage imaging. There is a major difference between those two probes is on their temporal dynamics. So calcium indicator are very slow, a voltage indicator is much faster. So if we generate a series of spikes and we look at them with the calcium indicator, there will be a signal piling up and lasting very long time, 100 milliseconds. While with voltage imaging, the indicators are much faster, so you can actually really optically resolve the spike. Okay, so can we do multi-target two-photon voltage imaging? Because again, we want to look at the spiking activity in scattering tissue from multiple targets. And then the problem is the following with respect to calcium imaging. So the calcium indicator, for what I was saying you before, they are in between, they are in all the cells. So you have a lot of fluorophores, you have a lot of signal. While with voltage indicator, they are sensitive to the membrane potential. So they have to stay on the membrane. So you have much less fluorophore that send light. First problem. Second problem, calcium indicator lasts very long, so you can sample your signal at a very low rate, let's say from 5 to 30 Hz, and you will be still able to follow your signal. But if I want to image this signal here, which lasts only few milliseconds, I need to increase my sampling rate. So we need to image at the kilohertz rate. Okay, so now this is really a problem because we want to image a very weak signal. On top of that, most of the the genetic encoded voltage indicator have a very low two-photon cross-section, okay? So we want to image a very low signal, a kilohertz rate. And this is why conventional two-photon scanning microscopy will never work. And so this is why it was very challenged to perform two-photon voltage imaging, two-photon voltage imaging. Okay, but again, we said we don't need to scan. We can do scanless two-photon voltage imaging, and so we build up a system. It's the very same as described before, where we can both have a generalized phase contrast or holography or simply large Gaussian beam. And now the idea is to place the entire spot on the cell, so we don't have to scan anymore. We can collect a lot of photons. Because you don't want to lose spatial resolution, now we need to use camera detection, okay? So we use the only indicator, or among the only indicator that works in two-photon is called JEDI 2P from the group of François Saint-Pierre, and which has been shown to work in two-photon. And so we started to look at what we could get, and indeed you see this is a cell. These are organotypic slice. Many cells express the indicator, and now we place our nice GPC spot, nicely temporally focused, and we can resolve the image, the signal only from that cell. We can even see the two membrane, and then you can generate a spike and resolve the fluorescent signal. So these are just analysis to assess the signal to noise ratio, the delta FRF. But I wanted just to show you that because now we are camera detection, so we can go very fast imaging rate. So these are again electrophysiological stimulation at different rate. We went up to 125 Hertz, and then in all cases, we can follow the spiking rate. Obviously, the higher there's, the better you resolve the spiking position here. And so with 1 kilohertz imaging rate, we were able to resolve a spike trains of 125 Hertz. And you can look at the spontaneous activity. So this is something I showed to tell you something more about the advantage of voltage imaging. Is not only the fact that you can nicely resolve the spike, is also that you can see sub-threshold signal. So you can see what happens also much before having a spike, which is something you cannot see with calcium imaging. And this is very important when you start to enter really and understand how signals are integrated in a circuit. So we need to see the spiky, but also the sub-threshold. And we wanted to take advantage of the fact that we can multiplex the light. So can we do voltage imaging from multiple targets? And the good news of this indicator that we found is that it can also be excited at 1030 nanometer. And why this is a good news? Because at this wavelength we have a laser that can deliver tens of watt, and also there are low repetition rate, so you can minimize the heating. And so we can multiplex the light, in this case 20 location, and having the temporal and spatial resolution necessary to resolve the spiking activity of those multiple cells. All these data were in vitro. You always have a referee asking for in vivo, so we added the in vivo data. So, but these are those to show that those indicators. Again, this is a field that is exploding now. So it probably doesn't tell you much now, but having a voltage indicator that expresses so well, it's a big advance today. And these are data. So again, same story. You can take your orthophoton image. You look at the cell. You see you have this nice expression on the membrane, and which you will also understand why scanning approach is not a very clever way, because you will have a very few light in the time of the scanning. While if you place a spot that illuminates the entire cells, you can be much more efficient. And this is what happens. So these are raw data, and these are the spontaneous activity recorded in vitro, also different depth. Obviously, camera detection is going to limit the depth, waiting for having nice demixing algorithm to go even deeper, but at least we can nicely reach the layer 2-3 of the visual cortex. So this is to say that one day, when we are not so far away from that, we could really now start to thinking of combining this fast sequential light targeting with a fast approach for high temporal resolution readout. If I have 5 minutes, I will show something on freely moving. Okay. Okay. So all of that is in vitro or in vivo. In vivo head restrained mice, meaning that you have a conventional microscope, then the mice is head restrained. The question was, especially for all these hippocampal things, all the cells, all the circuits which are implicated in navigation, in spatial orientation, or in fear, or in anxiety, you do need to investigate circuits in a situation where ideally the mice are free of moving in a certain space. So our point was, can we combine our approach so that we can do this all optical circuits manipulation in freely moving animals? And for that, we need to use a very specific class of endoscope, which are based on fiber bundles. Now I think everybody knows what I'm speaking about. So fiber bundles is a bundles of fiber. So you have 50 to 30,000 fibers. And why we want to have a fiber bundles? Because we want to transfer a pattern. Okay, so now I think this is more or less clear. We don't want to just send a point at a time, but we want to transfer a pattern. So these bundles are very nice because you can generate your patterns at the entrance of the bundles, and then this is transferred on the other part. And then you have typically a green lens or a focalizing objective that transferred exactly the same pattern in the mouse head. So the advantage of using this kind of endoscopy is that you can make an optical system as complex as you want. This will be on the optical table. And what reach the mouse is simply the fiber with this objective. So this can be less than 1 gram of weight. So you can really hope to not perturb too much the freely moving of the animal. So we did years ago this in single photo, but now I will consent you, I will show you what we did more recently. We call this system 2P Fendo. And so again, we want to do all optical. Okay, so we need to image. So we have a 2-photon laser that is coupled to the fiber. So to perform, to perform scan imaging, and then we have a second path going to a special modulator to generate multiple spot for holography. Okay, so which are the challenges to do photon imaging and photostimulation through a fiber bundle? This is probably now I'm really touching something you are very aware of. But anyway, there are 3 main things to be worried when we send light through a fiber. The first is the group delay dispersion. As we now know, in a pulse there are multiple colors. Those multiple colors won't propagate in the same way. So you can have an important broadening of the pulse after the fiber. This was a problem a few years ago, but now actually most of the lasers that you can buy, they have an internal compressor, which is enabled to perfectly compensate with this group delay dispersion. So we can have nice pulse coming out, fine. But there is another problem, more important, is the cell phase modulation. Is another nonlinear property of the fiber, so that actually becomes important at a certain power. So when you increase the power, you keep on increasing the power at a certain point, because of this effect, you will have an important pulse broadening, and this pulse broadening depends on the power. So means that at certain point, your two-photon response won't be quadratic to the power anymore. And so this has limited the number of light, which typically this was characterized by Fritjof-Helmchen a few years ago, is about 10 milliwatts for 80 megahertz rate laser. So why this is a problem? Because this means that I cannot couple more than 10 milliwatt per core, and so if I want to select enough photons during my scanning approach, we need to go slower. Okay, so we need to increase the dwell time. And so two-photon scan imaging through fiber bundle has been till now limited to an imaging rate of about 2 to 2.5 hertz. And so we wanted to see whether we could be more efficient. So do we really need to scan one core at a time, or can we shape the light so we can illuminate multiple core at the same time? And so again, because we illuminate multiple core at the same time, this will work. In principle, we can increase the time efficiency of about, if we increase the spot from 50 to 4 micrometer, we can get an illumination time 30 times shorter. So in principle, we could go up to 50 hertz. Yes, almost done. Okay, so and then holography is the same story as before. We need to have a large spot because we want to effectively activate the neurons. Okay, so now we have a double problem, both for imaging and for photostimulation. We illuminate multiple core, and so now we know the larger is the spot, the worse is the axial resolution. And so the question was, do we need to use temporal focusing also for fiber bundle? And in fact, what it turned out is that we don't, because there is this third effect that was called the intercal delay dispersion in the fiber, which is something that is related to the fact that to reduce the inter-fiber coupling, when people build up those fiber bundles, the diameter of the fibers is slightly different, so that you can reduce the coupling of the light. And because the diameter is slightly different, means that if you illuminate multiple core at the same time, at the other part of the fiber, they won't come up at the same time. There will be a small temporal shift between one to another. This was characterized by Vergniaud many years ago, and something we measured with Oricats, that building up an interferogram, he could really characterize the arrival time of the light at the exit of the fiber bundles. And in principle, this actually is of about, for this kind of fiber, this is a Fujikura fiber, is 2 picosecond per meter, okay? So what does it mean? It's the same story of temporal focusing. If I illuminate a large area, but in fact the core are never sending the light at the same time, it's like if you have an ultra-fast point scanning microscope, okay? So the light is coming out like this, so your resolution, no matter how many core you illuminate, will be always the one of a single core, which you can see here, okay? So no matter how many cores we illuminate, the axial resolution is always the same. So we have actually, again, decouple the lateral from the axial resolution just for an intrinsic property of the fiber. So we were able to perform, this is calcium imaging, in this case at 20 Hz. In this case, in the animal, we have only G-camp. So you see, we can perform fast calcium imaging with the animal freely moving. We can also go up, even upper. Now we are about 50 Hz. And quickly, we can also photostimulate, which is the major advantage of the system. So you can look at calcium imaging as before. You can choose cells, and you can activate those cells. You see the field of view is impressively stable. The mice is freely moving, so you can repetitively activate the same cell or the same group of cells. And going fast on that, now we have also changed the combination of fiber and objective, so this we can done in very large field of view. It's about 500 micrometer, so we have now access to relatively large circuits. Okay, this will really open the way to start to play with all circuits that are related to behavior that require the animal of freely moving. Okay, it's done. Okay, so all optical, so reading and writing is something is possible in a more and more precise way. I show you the most interesting application is to start to play with to see and play with the neuronal tuning, to see and play with the cell assembly and functional connectivity. And the more we advance with the technology, the more I hope new question will come up in neuroscience. And just to mention the people in my lab that are implicated with the data I show in this talk, and the collaborators and the financing. Thank you.